And now we have Aprinda from University of Florida. Aprinda, could you hear us? I kind of received an email from Aprinda that it seems that she's under tornado watch in an area in Florida that they have issues with tornado right now. But she sent us a video. Nikki, do you have the video? Can you kind of just broadcast the video?
Yes, sure.
Good morning from University of Florida. My name is Aprinda. I apologize that I cannot do this live because of the storm situation here in Florida. But I'd like to thank the organizer for the invitation for me to present in this wonderful webinar. So today I'll be talking about predicting functional connectivity changes in response to in-scanner TDCS using person-specific models. So this is just a brief background. Unfortunately, our brain starts to decline with normal aging process, and this include declines in working memory performance. And previous studies have shown that an acute or one-time application of TDCS can strengthen the functional connectivity within the working memory network that can help alleviate this decline. And this modeling study actually piggybacking a previously completed fMRI study. And in that study, we found an increased connectivity between the left DLPFC and the left VLPFC in older adults receiving an acute TDCS during working memory task. So the stimulation parameters that was applied was 2 milliamp for simulation for 12 minutes duration. So the objective of this modeling study is to first generate the individual current density maps or J maps in 15 older adults receiving this in-scanner TDCS paired with functional MRI. And we wanted to investigate the relationship between current density and observe functional connectivity changes between these two ROIs. So this is just the methods highlight. On the top here is showing the modeling pipeline that we use. So it's following the standard modeling pipeline, finite element modeling. The first step is segmentation. So if I can have your attention on this red box on the left side here, we take individual T1 weighted images and segment that into 11 tissue types. So they are white matter, gray matter, CSF, major arteries. So these are blood vessels that were visible in the T1 scan. Air pockets or sinuses. We separated the bone into spongy and compact. We also had muscle, fat, skin, and eyes. So the eyes encompass the sclera and lens. So in addition to the tissue types, so I forget to mention that the conductivity values that we use for the model is included here in underneath of each of this tissue. And in addition to the tissue, we also segmented the electrodes. So the electrodes are located in F3 and F4 locations. So the F4 here is the anode where we apply current of 2 milliamp, and the F3 is the cathode where we remove current. So I included 3 head models out of 15 to kind of demonstrate the variation of electrode location. So while we attempted to place these electrodes perfectly in F3 and F4 location, there's still some variation, not only person to person, but also with respect to the ideal location of 1020 EEG system for this electrode location. So after we segmented the models, then it went to volume meshing and then finite element simulation in COMSOL to solve for current density or J. And once we get the J maps, we apply this Gaussian smoothing filter. So this to mimic how the both images were processed. And if we can take a look at inside the blue box on the right side here, the J maps appeared a little bit blurry. So this is because we applied that smoothing filter. The 2 ROIs are represented in the circle here on the in the top on the overlaying the bold image. So the black circle is for left DLPFC and the red circle is for left VLPFC. So what do we find? We found there's a positive correlation between current density computed only in the left DLPFC and beta values, which is representing connectivity that was acquired only in active stimulation. So we did not find any correlation for computed J's in the left VLPFC, which is the second ROIs. as well as no correlation for beta values recorded during sham stimulation. So what did this tell us then? That maybe the large amount of current dose that was found in the left DLPFC ROI may be important in terms of eliciting this functional connectivity changes that we've seen between left DLPFC and left DLPFC. I'd like to acknowledge a large group of people who contributed to this project, especially Dr. Adam Woods, who's the PI of this project, and also the funding agency, NIH, NIA, Center for Cognitive Aging and Memory, and the McKnight Brain Research Foundation. Thank you so much for your attention.